A very good evening and welcome to one and all present here for the international webinar series DENTOPHARM on topic viral diseases and vaccine development. I am Dr. Ramya, part of Dental Education Unit and Senior Lecturer at Sri Balaji Dental College and Hospital. We'll be highlighting you about this program today. I would like to welcome our guest speaker, Dr. Murugabaskar Balan from Harvard Medical School, and I also welcome Chairperson Dr. Srinisha Ma'am, Registrar Dr. Bhuminathan Sir, Principal Dr. Jimson Sir, and Dr. Sumati Jones Ma'am, Head of the Department of Pharmacology at Sri Balaji Dental College and Hospital. Over to Sumati Ma'am to share a few words on today's webinar.
Welcome to DENTOPHARM webinar series. We are happy to have Dr. Murugabaskar Balan here. We welcome you, sir.
Thank you.
We are privileged to have sir as a visiting faculty in our department. We all know that reducing the infectious diseases will play a crucial role in population health as well as country's economy. So the first issue to be addressed for pandemic preparedness will be vaccine development. The sir will be explaining in detail about virus and vaccine development. Word to Ramya about sir.
Thank you, ma'am. I now warmly welcome our guest speaker, Dr. Murugabaskar Balan, scientist at Boston Children's Hospital and also an instructor at Harvard Medical School. He completed his bachelor in biotechnology at Anna University, Chennai. In 2010, he has obtained his PhD in biochemistry and molecular biology from the New Jersey Medical School, USA. He worked on studying the role of interferons in fighting viruses, infection, and utilizing interferon for cancer treatment. After PhD in 2012, he joined Boston Children's Hospital, part of Harvard Medical School, for postdoctoral studies. His research works are focused on identifying novel therapeutic targets in the treatment for cancer and vaccine development. At present, Sir has now joined as a visiting faculty at Sri Balaji Dental College and Hospital. It's a privilege to have you in our institution, sir. Thank you so much for accepting our invite.
Thank you. Okay. So thank you, Ramya. Thank you, Dr. Ramya. Thank you, Dr. Sumati. And first of all, I would like to thank the chairperson, the registrar, and the principal of Sri Balaji Dental College and the Bharat Institute of Higher Education and Research for inviting me as a visiting faculty, and I am very honored and happy to accept this position. And I would like to do more collaborative research with you all. So with that, I would like to start our talk today. So, which is viral diseases and vaccine development. So, I will be talking about the different viruses and the different viral diseases that we have known so far, and how the vaccines are developed for these diseases. I thought about this topic because we are just coming out of the coronavirus pandemic. And I thought it is the best time to discuss about the role of viruses in our life and how a vaccine saves life. So first, as usual, I will talk about the cells in our body, because we need to know how the cells in our body are affected by viruses. So as you all know, a cell is basically a cytoplasmic mass with a DNA material inside. And different parts of our body or different organs have specialized cell type. So basically all the cells are having a cytoplasm outside and DNA inside, but the structure may differ. For example, the brain cells have neuronal cells, which is elongated, and the lung epithelial or the heart epithelial cells, they are more in rectangle or in oval shape, and there are specialized hepatocytes or the liver cells. So depending on the organ, the cell's structure and shape differs. And there are blood cells, blood is a liquid organ, right? So they are floating in the blood, and it has red blood cells, white blood cells, which are known as T cells, B cells, macrophages, and dendritic cells. And so the liquid part of this blood is known as plasma. So we have to keep in mind all these cells, plasma, and the blood, so that we will talk about more about this when we deal with viral infection. So first we will know different types of viruses and bacteria. So as you all know, human cell can be seen in a microscope. Uh, like when you have a cell culture in a plate, you can see those cells in the microscope. And bacteria are very hard to see, and viruses are even much harder. So to see the virus, you need electron microscope. So in terms of the size, if you see bacteria would be say 0.5 micrometers, and viruses would range from 0.02 to 0.25 microns. So that means they're very hard to see. And to have an idea about the diseases caused by bacteria and viruses, so the common diseases caused by bacteria are cholera, pneumonia, tuberculosis, plague, and common viral diseases that we know are smallpox, measles, rabies, polio, dengue, chikungunya, and of course now the whole world knows about coronavirus. And to contrast from the bacterial and viral infection, malaria is caused by parasite. It is relatively larger than bacteria and viruses, and this is neither a bacterial disease nor viral disease, but it is a parasite. So as I mentioned, the viruses range from 0.02 to 0.25 microns, so the structure differs, but the basic structure is the same. So they have a very strong protein outside, the envelope protein outside, and they have a DNA or RNA genetic material inside. Say, for example, the polio virus is very small, and we have pox viruses, Ebola, rabies. They all have different structures. And now we talk more about coronavirus. So you can see it looks like the sun. So the corona means sun. It looks like a sun. That's why it's named as coronavirus. So when we talk about viral diseases, we only think of human being affected by viruses. No, even the plants are affected by plant viruses. For example, Tungro viruses destroys the paddy crops, and tobacco mosaic virus destroys the tobacco leaves. In fact, initially, even before scientifically viruses were known, scientists discovered the extract from this leaf as disease causing agent. So they crushed this leaf extract and infected to another tobacco leaf, and it developed diseases. So they named some particle inside this extract is causing the disease, but they did not know they are viruses at that time. But that's how they at least know the viral particles. And there are some viruses that infect animals. For example, the foot and mouth diseases for the cow. So these are all very much affecting the economy in the plant cultivation or the cattle development. So viruses not only affect human beings' life, so it also affects our economy through plant and animal infection. So here we should remember that viruses infect both human and animals, and some viruses, it can be named as one, say pox viruses. So pox viruses can infect humans, or there are pox viruses that can infect a cow. There are pox viruses that can infect horses, and so on. So as I mentioned before, all viruses have a very strong outer coat, so they cannot be destroyed very easily, and they have their DNA or RNA material inside, like the cell, but they don't have any machinery to survive by themselves. So that's why the virus is neither a living organism nor a non-living organism. So they can only grow inside a living cell. So when the viral particles infect a cell, so they make use of the machinery of the cell itself, the host cell, because they don't have anything to survive on their own. So they make use of the host machinery, and then they replicate, and then they rupture this cell, come out and spread to other cells. So that's how the viral life cycle begins. And viruses cannot survive outside a living cell, so they always need a living cell to be on the growth mode. For example, the viruses can be killed by soap water or disinfectant like ethanol, or sunlight can kill them, or heating them can kill them, or sometimes some surfaces can kill them. For example, copper surfaces can kill them. So these all depends on the physical damage to the outer envelope and then they cannot cause the infection anymore. So now we will see more about different viral diseases. For example, pox viruses. So this is very common, right? Smallpox, chickenpox. So, so the disease symptoms depend on the cell types that the viral infect. So each virus have a affinity for particular cell type in our body, and they grow only in that cell type, and then they replicate there, and then they, when the cell ruptures, they infect the other human beings or the neighboring cells. So, for example, the pox viruses, they infect the epithelial cells. That's why you get rashes on the skin. And then once these rashes, if you touch these rashes, the other person is going to get the pox viruses. So that's why you might have heard that people infected with pox viruses will be isolated. They have a separate bed and separate utensils and so on. So they are on the surface. So it's very easy to be contaminated to other person. And coronavirus, as we know, it infects the lung. So it enters through the nose, epithelial cells, and then the point of growth is lungs. And that's why it's so severe. It affects the airway system, right? So it's so severe based on its site of growth. And then the poliovirus. So this we know it will cause paralysis, right? So the site of infection is intestine. That's why it spreads through the fecus. So when through babies, the fecal matters, if you touch, it's going to contaminate the other person, or the fecal matter contaminating in the water. That's where these viruses spread. And the other side of growth is the nervous system down the leg. That's why it's going to call paralysis of the leg. And it's a very serious virus and deadly virus. And it can also affect spinal cord, and it can cause multiple organ paralysis. And rabies. Rabies virus are spread from dogs or through dog bite. So when the dog bites, the rabies virus in its saliva, it's going to enter our bloodstream, and the site of growth is brain cells. And that's why you will hear that people who are bitten by rabies dog, they also turn mentally disturbed. It's going to affect the brain cells. And Ebola viruses. So Ebola viruses, people say it can spread from bats or infected or bat bite or from human contact. And the site of infection is blood system. That's why people infected with Ebola virus, they vomit blood, and a lot of blood leakage or hemorrhages happen, and they vomit blood. And this you might have seen in the famous movie The Savadaram, like where this as a biological warfare weapon, and they're going to vomit blood. And it's a very serious virus too. And then dengue or chikungunya, they are spread through mosquitoes, and their site of infection is the lymph nodes in the joints, or the blood vessels in the joints. That's why people infected with chikungunya, they have muscle stiffness or bone ache or severe bone pain. And this is a very recent outbreak, and you might have heard of chikungunya during monsoon season because of the mosquito spread. And sometimes people ask me, like, why you get chikungunya these days, not like 100 years before? Yeah, that is chikungunya virus as such was existing or is existing for millions of years. But the problem is, it started to infect someone in the African region from the forest when they destroy forest and they go for hunting or whatever, and these viruses spread to that individual human being, and then he traveled to some other place. And in fact, people trace the spread of chikungunya by tracing who are all infected, and then it came to Maldives, and then from Maldives somebody traveled by ship to India, and from there it started to spread to the whole nation. So as you know, that's why even the videos are called viral. It spreads like anything. So once you have the medium of infection and the source of cell that it can survive, it will keep spreading like fire. So here we see that different viruses have different affinity for different body cells, and they take advantage of that cell type and cause disease. So when these viruses infect our cells, our immune cells or the blood cells can fight the viruses. So once, say for example, in coronaviruses, in the case of coronavirus, if these cells are infected with virus, so what will happen is our blood cells will sense this viral infection. So there are some viral particles that our blood cells can sense, and then they will release interferons. So interferons are named as interferons because they interfere with the growth of virus. So they can release interferons and then they try to kill the viruses. This interferon, by acting on these cells, on the human cell, it can destroy the whole cell, including the virus. But the viruses are smart as well. So they secrete some protein that can be counteracting against these interferons. Those are called interferon antagonists. So there is a fight between our immune cells and the viral infected cell, and finally, who wins? Then the disease will be manifested or it will be suppressed. So in the course of fight, there are B cells in our body that recognize this infection and they produce antibodies against this virus. So in the future, if you get the same viral infection, so these B cells are ready. These are called memory B cells, and they're ready to fight this virus immediately, and it can clear the infected cells within hours to days. So these are always in circulation in our body after our first infection, and that's why when you take the plasma or when you centrifuge the blood, all the cells will be down and only the liquid portion of the blood, which is known as plasma. So this plasma contains all the antibodies against this virus, and that's why you might have heard about the plasma therapy during coronavirus pandemic, because those who recovered from coronavirus, their blood has this antibody against the coronavirus, and you cannot give the whole blood, it's going to cause reaction. So you just purify the plasma, that is the blood portion without the cells, and you inject to the person who is affected. So now these antibodies in this plasma can help that infected person to fight infection. So depending on this time of fight between our body and the viral infected cell, it can manifest in within 1 or 2 days. Say for example, the common cold or even coronavirus, within 1 or 2 days you feel the symptoms. So pox viruses in 10 days. So polio can be within few days, and HIV, it can be in our body for 10 hours, 10 years, but it won't be manifested. So people living with HIV, they are positive, but they are dormant. So it can manifest anytime. When this fight is taken over by the viral infected cell, then it can manifest as disease. So we have seen that our body can protect the virus after infection, the first infection. So that is the concept of vaccine. So the antibiotics, for example, are drugs that can interfere only with the growth of bacteria, but not virus, because bacteria can grow by themselves because they have all the cytoplasm and other proteins for their growth, and these antibiotics will stop the protein synthesis in this bacteria and they kill them. But as I said before, viruses, they don't have any machinery for themselves, so they rely on the host cell. So you cannot kill the virus with the antibiotic because you are killing our own host cells. So there are class of drugs called antiviral drugs, say for example hydroxychloroquine. So what they do is they interfere with the maturation of the virus. So when the viral enters, some drugs can block this entry point, or some drugs like hydroxychloroquine can block this vesicle release. So then they won't spread to the neighboring cell, and they're contained within this infected cell, and then they will be cleared by our own cell, like by phagocytosis and other processes. So the antiviral drugs can interfere with the viral entry or replication or the maturation and the exit from the cell. So those are chemical strategies. But we have seen that our body has memory for viruses. Say, I'll give an example with the pox viruses. So as I mentioned before, pox viruses can be infecting humans, or some type of pox viruses can infect both animals and human. For example, cowpox virus and smallpox virus, which is called variola virus, and it can infect human. So this vaccinia virus can infect both cow and humans. So what they found in old days is they don't know about the concept of virus or viral particles, but they know that people working with the cow, like the milkman, or people working with the horse farm, they don't get smallpox infection, even though it was very deadly disease at the time. So they found that only for people working with the cow and horse, they don't get this infection. So then that was the Jenner's experiment. He found that some particles in this cowpox lesions or the horsepox, they are protecting these individuals from smallpox. So what he did is he took these cowpox lesions and inoculated them to the human and waited for 2 months and then gave back the smallpox lesion. So there's no, they don't know, they don't purify virus or anything. You just take the smallpox lesion from somebody infected and give it back to the person. So whoever got inoculated with the cowpox lesion or horsepox lesion, they did not get this virus back. So then he named this concept as vaccination, because vacca means cow in Latin. So that's how the whole concept of vaccination came. Even before scientists discovered the viral particle or we could know the structure of virus, it's just from the lesions he found this vaccination strategy, and the vacca comes from cow. So then people wanted to develop vaccine for polio at the time. So this was successful for few years, like 50 years, and then scientists wanted to develop vaccine for polio, but they did not succeed. As you can see here, we were lucky because this pox virus infect both human and cow. So one lesion from cow can save humans from smallpox virus, but we don't have a polio virus that will infect non-human beings, so that we can take those extract or lesions to inject back to humans to avoid polio. So that was the problem. So we don't have other system where we can make use of that system to protect from this specific virus. In fact, in olden or ancient India, China, what people do with the smallpox is, say people recovering from smallpox, right? So they take the lesion at the end of the disease. So at the end of the disease, now we know that the viral particles will be very less. So instead of say 10 power millions of virus in one lesion, they may have maybe 1 or 2 or 10 particles in that lesion when they recover, because smallpox is deadly at the time, but somebody is recovering from that, their lesion will have very few viral particles at the end of the recovery. So then they take this, they collect these particles and then they inhale these particles through nose. So by this way, they are inoculating themselves with the low dose of smallpox virus directly. So that was also a vaccination strategy before Jenner in ancient India and other nations. But Edward Jenner was the one who named this vaccination by an experiment. He proved this with an experimental cowpox vaccine. So the key for producing vaccine is to grow them in the lab. So I mentioned that polio cannot be grown in other non-human non-human organisms. That's why we could not get the polio vaccine. And then later, like 1950s, scientists discovered the virus. They could see the structure of virus in electron microscope, and they know what are the cells types that are infected by specific viruses. So now the strategy is to grow this virus in the lab, or like say fertilized egg. Like the fertilized egg, it's a kind of incubator because it's growing, right? It's a living incubator. So you infect this virus into this egg, and they start to multiply in this growing embryo, and then you destroy this egg and collect the viral particle from centrifugation. So when the technology or the science developed to grow the lab in virus on a cell culture plate, you infect say human kidney epithelial cells with these viral particles, then you can produce tons of viral particles. Or you grow the virus in the egg and you can produce tons of viral particles. Then you take these tons of viral particles that you produce and then you kill them, or you heat inactivate them to kill them. So then what you have is viral particles that are not active anymore. You may have just the envelope, little bit of envelope, on some DNA material, so or the RNA material or the protein outside. When you kill them, when you destroy them, and then you inoculate this into the human beings. So now that mimics a viral infection. So instead of giving a cowpox lesion for smallpox, we are taking, say, for the polio virus grown in lab, and then you destroy them and then give them a little bit of these viral particles as a vaccine. So that was the initial stages in 50s and 60s. Scientists developed this way to grow viruses in the lab, and then we got vaccine from that killed viruses. So what will happen when you give this shot into your body, like small amount of viral particles? So all your blood cells will have a memory of this small amount of virus. So when you give a small amount of polio virus into your body as an infant, so then all your T cell, B cell will recognize them, and they're ready to fight when there is a real polio virus infection in the future. So that is the strategy for vaccination. So in the 70s and 80s, so you will see for the 70s kids on their arm, you will see two scars. So one is BCG. This is for a tuberculosis vaccine. Even though it is a bacteria, it is the same concept. So bacteria grown in lab, you kill them and then give them back to prevent TB. That was also popular in 50s. And then when the smallpox were grown in lab, because we cannot rely on the cowpox lesion for long. So when the strategy to grow smallpox viruses in the lab was established, we could get tons of smallpox viral particles, and you inactivate them and give little bit of very little dose. I mean, if you give more, you're going to infect that person with smallpox. So you give a very little bit of smallpox on your arm, and then it left a scar, because those days the inoculation was so with I think it was with the heated needle prick. So that's why it left a scar on them. And then after 80s and 90s kids, they will have only one scar on their arm. That is only for BCG, because the smallpox was eradicated. Because when you give vaccine to all of the population, then the virus has no place to grow, and then eventually it is destroyed. So that's how the smallpox was eradicated with a very aggressive campaign for example, in India in 70s, and it was officially announced eradicated from India in 77, and from 79 it was eradicated from the world. And now we are on the path to eradicate polio, even though we are officially eradicated, but there are some cases here and there. But we should still keep giving the polio vaccine. And now, as I mentioned, the technology has improved, so we could give them as an oral drop or as an injection. So both are or heat inactivated or killed polio viral particles grown in lab, and you give them to the infant. So then they are protected from future polio infection. And our technology has even advanced. So, for example, here it may not happen with the one dose. So when you give one dose, you may have a little bit of memory with the B cell and T cells, but it may not last long. So that's why you give booster doses in a span of few months or few years, so that you reactivate those memory B cells to be very much active. So this you can measure the extent of vaccination by measuring the antibodies in the plasma. So you can have a specific antibody test like ELISA, and then you can measure the antibody against this specific smallpox viral protein or coronavirus protein in the plasma. And then you can measure, like, say, the coronavirus spike protein, and then you can measure how much memory is in your blood. So those days, the rabies viruses that was cultured and given to people, like you might have heard, like when there is a dog bite, we have to give 21 shots around the stomach. So why stomach, not the arm? Because you are giving 21 shots, right? You cannot puncture 21 in your arm. So the stomach has a lot of fat, so it can bear 21 shots. That's why it was given around the stomach. But now we have with the technological advancement, we have rabies viruses that are grown in different strains of rabies viruses that are more strong in eliciting immune response. So now we can be done with 3 or 4 shots, and that too in arm. So no more stomach inoculation. So then coming back to the coronaviruses, so these are called zoonotic diseases. As I mentioned about chikungunya, it came from deforestation, contact with animals, or zoo means animal. So similarly, there are thousands of viruses still that we don't know. Maybe millions of viruses in around the world in different organisms, but only 250 are known to cause human diseases. But all out of those thousands of viruses, they have potential to cross to human. So those are called zoonotic viruses. Say, for example, from uh, pig or swine or chicken or bird like swine, or from insects, or from monkeys, from cow, from different animals. They have different animal disease causing viruses. They have the ability to spill over to human because of the mutation. Say when somebody is infected with the virus from swine, it may not cause disease for this human. But say they have a mutation that is specifically modified for human, it can cause infection. So that's why it spill over from animal to humans. So these are called zoonotic diseases. Even HIV, there is a theory that it spread from eating chimpanzee meat. From that blood products, it spill over to humans, and from then human it got infected to other humans. So similarly, bird flu or swine flu, it has the ability to spread to humans. So it's not like now we only talk about COVID-19 pandemic, right? But previously, there was several zoonotic diseases that got spilled over from animal to human. Say Ebola from bats, Nipah virus. In 2002, there was SARS, severe acquired respiratory syndrome causing virus. So that's why we have SARS at the time 2002. Then bird flu, swine flu, then MERS from Middle East respiratory syndrome from camel. Like when people are in close contact with camel, some camel viruses can spill over to humans. And now we have ongoing pandemic, the coronavirus. So most likely it got spilled over from bat. That is the theory. There are conspiracy theories that it might have been spread from the lab. It is possible, but right now the evidence says it is less than 1% chance for that. Mostly it is from bat, because people sequence the virus from bat. They isolated the viruses from that area bats, and they found sequences similar to what we have right now in around the globe, the coronavirus. So if we talk about the coronaviruses, as I mentioned, there is SARS coronavirus, there's MERS coronavirus, or SARS-CoV-2, what we have now, and then there are common cold coronaviruses. So this is very important. Like we have coronaviruses with us all the time, but when they get genetically modified, so they become more affinity towards human beings, even than the bats or camel, which is the natural source of infection. They get mutated on the spike protein. This is the spike protein. So as I told, the viral entry is the important step. So they have a new mutation on this spike protein gene, and now they can infect human beings very strongly. So that's the problem with this coronaviruses. Otherwise, we always have coronavirus. They just cause common cold 2 or 3 days, and then it's gone. Our body can fight it. But now when they become more mutated and more strong, our body immune system cannot fight them. So the first step towards that is to detect the virus. As I mentioned, the viruses have RNA particle inside. So what you do is you take the swab and then you isolate RNA from that. All we are talking about microns. So we cannot see anything in the swab or in the saliva. We cannot see anything. But once you have a kit, it's all molecular biology kit these days. I mean, those days the viral purification was very tough, but now we have all the kits to purify virus. And then you extract the RNA from them. You do a reverse transcriptase PCR or the real-time PCR, and then you see the specific gene in your sample for the coronavirus. So then you can detect immediately after infection. Or you can wait for a few days and then take the blood sample or the swab again, and then you drop that swab in a liquid, and then there is a kit. where you have an antibody here or the peptide here. So now when you drop this virus, so this antibody here coated with the antibody can recognize this virus outside of the virus, and then it can give a signal like a colorimetric signal, a color-based signal. So these are the ways to detect the virus. That's the first step in fighting the virus. We have to differentiate what type of virus is it, whether it's really coronavirus. So you can go for a specific region of the virus in the RNA, or you can go for the spike protein on the outside. So all these are in microns that you have to imagine. So then how to fight coronavirus? So the same strategy we are using now. So the antiviral drugs, but those are not very effective because they are so aggressive that you have to increase the dose, and by increasing the dose, it may be very tough for the. It may be very it may cause very much side effect for the humans itself. So the antiviral drugs are not that much effective against coronavirus, although it can give some protection. And then we have there is a revolution in the vaccine strategy after the coronavirus, because I mentioned like previously, what we have is just you grow the virus in the lab and then you inactivate them, attenuate them, or kill them by heat or chemical, and then you give back small amount of that same virus. So that was the strategy before for a long time. And now what is the strategy is you synthesize this mRNA directly, or you synthesize this peptide directly in the lab. So you don't need any viral particles. You know the sequence of this. You just synthesize that RNA in the lab, which is called mRNA, or you synthesize this peptide, a little bit portion of the spike protein, that is called peptide, and then you mix this mRNA with some adjuvant, which can stimulate your immune system, or you mix this peptide with some adjuvant and inject them directly. So, or what you do is you clone this gene into different virus, like a not very harmful virus, or not very much highly replicating virus. You just clone them into adenovirus, some part of the coronavirus gene into this, we can say non-disease causing virus. So I will give an example. So what we have is, this is the co-vaccine strategy from Bharat Biotech. So you can grow the coronavirus and then kill them and give a little bit of the coronavirus back to human. This is the Bharat Biotech co-vaccine, and this is the Pfizer or Moderna strategy, which is not approved in India, but US and other countries, they approved this, because you just take the mRNA, no virus inside, and then give it back to the humans. So they make this viral protein inside our body, and then when the real viral comes, they're going to make this protein. So then our body system can sense this when there is a real infection. And then we have the other one, I forgot the name. So we have Bharat Biotech, and the other, I forgot the name. So that's very common, right? So that is also from that is very widely given in India. So that is adenovirus-based vaccine. So you clone your gene into the non-virulent virus and you inject them back. So these are the three viral strategies that were employed to fight coronavirus. So mRNA strategy, live vaccine, and a different virus, non-virulent virus, carrying the genes of coronavirus. So there are also reports that a bacterial vaccine, people who were given bacterial vaccine, has some protection against the coronavirus, but that is not proved completely. But this could be coming from stimulation of our immune system. So any vaccine you give, say BCG is a tuberculosis vaccine, but it activates the immune system. So once your immune system is active, it can fight any infection. So maybe people who got BCG, they can fight coronavirus, but that is not proved. So the vaccination is the best strategy for any virus. We have seen that polio viruses were eradicated, and smallpox viruses were eradicated. And now with the coronavirus, when everybody is vaccinated, then there is no place for the coronaviruses to spread, and then it will be eradicated. But the viruses are also smart. So they change the spike protein often, and that's why we have different variants of coronavirus. So when you have different variants of coronavirus, you are targeting only some gene, but when the gene is modified already in the virus, it may not be effective. It may be a little bit effective, but not that effective. So the best strategy is, I would say, in my scientific literature review, the Bharat Biotech vaccine strategy is the best, because it is the whole virus that is given to the body, so it can develop antibody against many regions of the virus, and it can induce strong immune reaction, and it can have lasting protection. So now let's talk about the viruses and cancer. So viruses not only cause viral diseases like fever or cough, cold, or like lesions, but they also can cause cancer. For example, hepatitis B virus, it can infect liver, and once they start to rupture the liver cells, it becomes cirrhosis, and it can eventually become a hepatocellular carcinoma. And then there is, I'm sorry. And then there is human papillomaviruses that can infect head and neck or the oral cavity, and they can form oral cancer, which can be of interest to students of dentistry. So human papillomaviruses can infect oral cavity, and it can cause oral cancer. And there are other commonly cancer-causing viruses or Epstein-Barr viruses. It can mostly for immunocompromised patients. So these viruses infect, say, people who are undergoing transplantation, like liver transplant, kidney transplant. They are on immunosuppressive drugs, and they are prone to this viral infection. And then these viruses, once they attack the cell type and they rupture the cells, they cause lesions, and then it can turn to cancerous lesion. So there are viruses that cause cancer that we have to remember. But the same viruses can be used for cancer treatment. So as I mentioned, like the other viruses that are non-virulent viruses, so they are inactivated, say adenoviruses or lentiviruses. They are viral-like particles, so meaning they can enter the cell, they can grow in the cell for some time, and then they will be inactivated by themselves. They make some modification in their genome, so they are no longer virulent. They can enter and infect, but not cause disease. So those viruses, you can use them as a carrier for your gene of interest. So we have seen that the vaccine strategy, where we load the coronavirus gene into these adenoviruses. Similarly, we can load some of the good genes to kill cancer into the viruses, lentiviruses or adenoviruses, and infect this cancer patient. So then what will happen is, specifically, that you infect the T cells, you purify blood from the patients, you take the T cell and infect these viruses carrying good genes to fight cancer, and then you inject these T cells back to the body. Now it can fight the cancer by itself, because you have loaded the system with cancer killing genes. If you remember in my previous lecture, I talked about tumor suppressive genes. So you engineer these T cells with the tumor suppressive genes. Say, for example, T cell receptor, it can be active more to make these T cells kill the cancer cells naturally. So that's one use of non-virulent virus to introduce novel cancer treatment. And also there is a concept of cancer vaccine. For example, the same non-virulent virus, you load them with some of the good genes. Say, for example, GM-CSF, granulocyte macrophages colony stimulating factor. So what this does is, the GM-CSF in the environment, it can produce a lot of growth factors that will attract immune cells to kill the cancer cells naturally. So there is an approved cancer vaccine for melanoma or the skin cancer. So what that do is, you infect the non-virulent viruses carrying this GMSF, and then you can inject on the cancer cells directly. So it's going to release a lot of factors, and then it attract immune cells. Then the cancer cells can be killed naturally. So even if this goes to the normal cells, it does not harm itself, because it's not this. This is a non-virulent virus. It's not going to cause any damage to these cells. And even if they secrete the factors, those factors are good factors, so it does not harm the normal cells. And even BCG vaccine is used to fight bladder cancer. As you know, the bladder cancer is in the bladder area, right? So it cannot, it's not accessible for any drug. So there is a study that, and even it is approved, the BCG vaccine, even though it is a bacterial vaccine, which is a heat inactivated tuberculosis strain, you inject them in the bladder area, stomach area. So then there is a global immune response in that area, and then it can fight cancer, meaning the immune cells that are activated through this vaccination strategy can go fight the cancer cells. So the cancer vaccines are the next step to make use of the virus to the advantage of human beings. So far we have seen, in conclusion, we have seen that different types of viruses, say animal viruses, plant viruses, and viruses that causes diseases in both humans and animals, and the vaccination strategy. So VACA means cow, and Jenner introduced this concept of vaccination using cowpox to cure smallpox. And then now we don't need to grow viruses in the lab. I mean, the growing viruses was the first step in eradicating many viruses from the world, like including smallpox and polio. But now the new strategy is taking just the mRNA or protein from the viruses, that too not from the virus directly, but artificial mRNA or artificial peptides, and make use of them as vaccine. And then we have seen how viruses can cause cancer, like hepatitis infection or HPV infection can cause oral cancer. And then we have seen how to use the non-virulent viruses to kill cancer cells. And then of note, we have to be very much aware of the zoonotic diseases, because we have seen how much we are affected through the coronavirus pandemic, which got shifted from animals to human. So there are people working on a global virome project to sequence all the viruses from known animals, say bats, because bat harbors so much of viruses inside them, and scientists want to sequence them to see if there is some sequence that some protein that is best fit for humans by modeling. Like, you can model the protein and see whether it can bind with high affinity to our protein on the human cells. So there is a global virome project going on, an Eco Health Alliance, to prevent animal misuse or forest deforestation. And then there is a Zoonosis Day observed on July 6th to make awareness to prevent future outbreaks. So that's all I have for the viruses and vaccine development. I'd be happy to take any questions. Thank you all.
Thank you, sir. You have explained clearly about viral vaccines, vaccine development, and it was really useful to the students. We have only few
I have a question. I forgot the other vaccine. What is the other vaccine? Covaxin and
Sputnik.
No, there's another one in India.
Covishield.
Covishield. Covishield is the one with the non-virulent virus,
Yes.
but with the coronavirus gene inside. So that's
Yes.
what I forgot to mention to students.
Covishield, sir.
Yeah, Covishield. Yeah.
Covishield. Sir, we have only basic question like this. How can we detect genotoxicity of vaccines?
Like, there is no genotoxicity. You mean it can modify the genes later? I mean, there are some anti-vaccination groups that say that it can affect some neural genes, it can cause some developmental disorders, but that is not known so far. But I would say the new strategies needs more study, like say mRNA vaccine or the Covishield, because those are a different strategy from the regular vaccination strategy. So the regular vaccination is nothing but you are giving a small amount of the same virus. So there is no harm in that. That is being used traditionally, even before people know about virus or viral particles. But the new strategy needs more testing, definitely, yes. Whether there is any harmful effect when you introduce the mRNA to our body is going to alter any genes. But so far, there is no data supporting that it's going to have any ill effect. So there is no data supporting that.
Okay, sir. Do we have animal-free test methods for development of vaccines, sir?
Yes. So, like I said, the polio previously it was used on monkey cell line, and previously people grow them in some other animals that can harbor this growth of virus. But right now we have all human cell line. So what we have is modified human cell line, say human embryonic kidney, epithelial cells, and then you infect one dose of virus. It's going to produce tons of it. Then you collect from that supernatants. You purify the virus by centrifuge. You kill them and just give a little bit of them. Yeah, we don't need a animal source anymore.
Okay, so we don't test with animals
Yeah, and
nowadays.
even for this mRNA strategy, we don't even need any cells. We just have to synthesize this mRNA in the lab chemically, and then mix them with chemical adjuvants like alum or other adjuvants, and then you give it back to the humans. Yeah.
Sir, few words about edible vaccines, sir. Advantages and
That's the oral
disadvantages.
route. Oral route. It's a polio, right? For
Yeah.
example, polio, the site of infection is intestine. That's why you give oral. It's more effective. And for say coronavirus, we cannot theoretically you can take it, but it won't elicit immune response because their site of infection is not the oral route. Theoretically, you can inhale or you can even buccal cavity you can give, but the best route is arm. So then you activate the whole blood system.
That's all then, sir. These are the
Okay.
basic questions from our
Okay.
side. Thank you, sir. Thank you
Thank
once
you.
again.
Yeah.
Over to Ramya again, sir.
Thank you, sir. Thank you so much for explaining the hierarchy of the viruses and the vaccine development and the etiopathogenesis from human to animal and so on. Indeed, you are a great teacher, sir. Thank you so much for explaining us in detail.
Thank you. Thank
I
you.
would,
Bye.
thank you, sir. I would also like to thank the institution, Sri Balaji Dental College and Hospital, Dr. Sumati Jones, Head of the Department of Pharmacology, for conducting the webinar series. And I also thank Dr. Jimson Sir, Principal, and the organizing committee, Dr. Satish and N.T. Murli. A special thanks to Doc Mode, Dr. Nishat Sheikh and Angelin for the technical support.
Okay.
And I also thank the audience for participating. Thank you.